Please.
Thank you very much. I don't know why this came out upside down, but it doesn't matter.
Fantastic.
Yes, just to start a little bit of overview of all the projects. We have been involved in genomic sequencing for more than 20 years, actually. And at the very beginning, there was the East genome that I still remember. We were paid by base. Every base that was sequenced, we had something like 6 euros, 3 from the European Union and 3 from Italy, 6 euros per base. And we heard earlier on that Nature, at the beginning of the year, in an editorial, said that this should be the year of the $1,000 genome. So it's very exciting that things are going so well and the technology has developed so quickly. The most recent projects that we have been involved with are just there at the end, and some of them are very challenging. I would like to mention the wheat genome, for instance, that is a genome that is a huge genome, 17 billion bases, and Italy is responsible of the sequencing of one chromosome, chromosome 5A, that alone is about 800 million bases. So it's bigger, for instance, than the grape genome that we sequence, that we're still finishing off now. So there is a lot of activity going on, and certainly the $1,000 genome is a very interesting news. But look at this. This is a report that came out very recently, and you cannot read very well here, but I can highlight this point here. $1,000 genome, yes, that's very nice, but $100 million interpretation, because this is what the situation is. Now, to produce the raw data is reasonably easy, and is becoming easier and easier, but to make sense of the data is still a very big challenge. And of course, $1 million doesn't mean that it costs $1 million. $1 million means that you cannot do it at all, or that you can do it very partially, because what we can get out of the information from the genome sequence is still very, very partial. So we heard earlier on a lot of very interesting specific topics. I would like to move a little bit more on the technical aspects, and I will focus more on, okay, very nice, we can produce the raw data for $1,000, but what can we do with this data? And essentially, there are 2 or 3 steps to do a genome sequence. First of all, we have to produce the raw data. That is still very important, and it's not always so easy, and it's not always $1,000. Believe me, it is a bit more expensive, but it's true, it's getting cheaper and cheaper. The second problem, that is still quite an important challenge, is to put together all the sequencing. So to do the assembly process, and to go from million and million of reads into the finished genome sequence. And of course, the third challenge is to make sense of the genome sequence. So I will go over this, what we are doing also in Padova, to try to answer some of these questions. So bioinformatics, we have a lot of activities in the bioinformatics, and that is supporting also the sequencing that we are doing. I was showing you before, most of the sequencing is done now with either the 454 that we have in our spin-off BMR Genomics, or with a solid platform that we have at the crib itself. So we use these 2 technologies, and most of my talks will be about these 2 technologies, to put together these 2 technologies to produce a good genomic sequence. And of course, both technologies and ourselves can be used for RNA-seq technology, DNA resequencing, so genome resequencing, and the novo genome resequencing, that is really a very interesting and challenging approach to also resequencing. Now most people say that even human genome resequencing is not good enough, because if you try to map your reads onto a known genome, you have only a partial view of what you really have. So most people prefer to think about the possibility to do a de novo resequencing also on genomes that are already known, because you have an unbiased version of your new genome, and then you can compare this unbiased version to your reference sequence. Okay, let's go on. Well, I want to show you just one slide about RNA-seq, because it's really nice. This is done with the solid, and the nice thing is that you can do strand-specific reads. So in this case is the grape genome. This, the green is the leaf, and the red is the root. And you can see, for instance, this predicted gene here is expressed in this tissue, in the leaf, but not in the root. This other one is expressed in the root and also in the leaf. And also you can see very easily some of the exon, like this exon here, that is expressed in the leaf, but is not expressed in the root. So with this technology, you can really have a very good indication of the genes. I was telling you before that one of the problems is to make sense of the data, and RNA-seq is certainly a very good approach, because the coverage is so high. I can just tell you this, with one single tissue, with the two tissues together, root and leaf, in the grape genome, we managed to cover about 60% of the predicted exons. So it's really a very good and powerful method to do gene prediction. That is the first step that you want to do, the first analysis that you want to do when you have the genome assembled. But this is all I want to say for the transcriptome, and let's go instead on the analysis, on the assembly of the genome. What are the problems that are making the assembly of the genome still a little bit difficult? Though over the past 2 or 3 years, things have been improved quite a lot. Well, the first step is to align the reads. Our group we ourselves did some work in this. We developed 2 programs, one based on graphic processors. The graphic processors are really incredible machines. They are very cheap. They have more than 100 single cores inside, and you can parallelize very, very easily some of the processes. So instead of using them as graphic processor, we use them as computing CPU for data computing. And we still use this for some of the application, and this is another program that is very good, and we use it very, very much to align sequences. Why do you need a special program to align this huge amount of sequences? Because even if you should use BLAST, just to tell you, and you should align 100 million or more, hundreds of millions of reads, the time would be so long that would take months before you could complete the project. So you need some new algorithm that optimize the use of the memory, optimize the use of the CPU, and allow you to speed up the process. Now, compared to just a few years ago, we can have much bigger computer with a lot of available memory, and we can use this facility to develop a program that can do this job. This pass, essentially, what does it do? Well, this is the graphical interface. I'll go over very quickly. Essentially, once it finds a potential point of alignment, it tries to extend this alignment in a very simple way. Essentially, what we do is we perform all the alignment of all the possible sequences of 8 bases. You know, there are 65,000 possible sequences of 8 bases. You know that an enzyme 8 cutter cuts about every 65,000 bases. Well, we can align all the sequences against themselves. So you have 65,000 sequences aligned against 65,000 sequences. Why do we do such a crazy thing? Because we do this job only once, and After we have done that, and we have done this also with 9, also with 7, what we can do is instead of extending the alignment like Blaster does, one base at a time, we can take big words of 8 or 9 bases and check if the whole word is compatible with the alignment. So essentially what we do is to speed up the process because we look for very significant words of 8 bases rather than single bases. But think that if you do all these alignments, you need 4 billion results. So we have 4 billion results of all the possible alignment of 8 bases. It's quite incredible to think that such short sequences align against themselves, produce such a huge amount of data. But 4 billion is just 4 gigabytes of memory, that for a computer is almost nothing. And therefore we can very easily put this in the memory and proceed much, much faster with our alignment. This is a very important thing, because if you cannot do this very quickly, then all the process is slowed down immensely. So, when what I would like to talk is about this problem. When you do de novo sequencing, essentially, or in general genomic sequencing, you have 2 important steps to go through. The first step is to produce contigs that are produced by overlapping sequencing. So the computer tries to overlap as much as it can the DNA sequence to extend the contigs. So this is one step, very important. The product of this step are the contigs. But there is a second step that is also extremely important, and it is a little bit, I think, not optimized as it should. That is the scaffolding process. So try to put all the contigs together. And one of the best ways to do this job is to consider the 2 ends of an insert. This was done even with Sanger sequencing. You could have, for instance, 3,000 basis inserts, and you were sequencing one sequence on one end and one sequence on the other end. And this information, so the fact that you know that those 2 reads must be at a certain distance that correspond to your library, is a very important information. And this information can be used, of course, to help the assembly. With the low coverage Sanger sequencing, this was not maybe so important because the coverage was too low. With the new generation sequencing, it becomes very, very important. I was showing you before, this is a typical result that you can get. You get hundreds of millions of mate pair, meaning that in our case you have 50 bases plus 50 bases that you know that are at a certain distance in the genome, for instance 10,000 bases, and you have maybe 100 million of them. So if you consider that you have these 2 sequences that are about 10,000 bases apart, you can imagine an arc that is merging, that you know that those 2 sequences are joined by this arc, and they must be at a certain distance. Right? If you have this kind of numbering, 100 million with 10,000 base long insert, the actual physical coverage, so I mean, each single base will be covered by hundreds and hundreds of these arcs that are putting together, keeping together the sequence. So essentially what we can do is to try to map all our mate pairs onto the contigs and try to do 2 very important things. One thing is to find the intra-contig, so all the mate pair that map inside the contig, and we can use this to confirm and to validate that the contig are good, because if there was a recombination event or a misassembly of the contig, then we would find that there is a problem. And the second thing that we can do is to take the intercontig mate pair that should help to put together the genome. So this is essentially what we do. This has been done for the tomato genomes, and now we are trying to do a similar process but a little bit more difficult because the size of the genome, also on the chromosome 5A of the wheat. So this is more or less what we do. We start with the 454 reads that are very good to produce reliable contigs because they have long reads and they're reasonably good quality. We use Nubler to do the assembly. Nubler is the program of the Roche 454. And then we use this contig to map the mate pairs of solid, and eventually we can improve the contig and scaffolding. So this is the process. We can use the intra-contig mate pair to do a statistics, so we know that the insert length of the library is within a certain range, and this is very important to apply a proper statistics to evaluate if all the little arcs that are over a certain position are compatible with the length of the library. So, at the end, what we get is a lot of arcs that we try to make a consensus of every possible joining of all possible contigs, and we get something like this. So we get, of course, very often you have tens of thousands of contigs that you have to put together, and you have indications that certain contigs are merged to other contigs, but not always that one contig is merged to the next. Sometimes they are very long links. So the program that we have developed allows us to go from this condition to this condition and to get a scaffolding that is much better. So just to show you this, we try to improve the assembly obtained just with the 454 reads, using also the solid reads. And so you see, the Nubler, the 454, leaves gaps. Instead, with the consort, we can insert some extra little contigs in these gaps and fill the gaps, because there is all this indication that tells us that those contigs should be there. Also, we can estimate much better the distance between contigs, so the gap size, because we have much more information, because, as I said before, you have hundreds and hundreds of arcs that go over. So this is another case. So we were very happy. Of course, some of how do we know that our assembly has actually improved the Nubler assembly? We could check all this on some of the known back that had been sequenced before, so we could confirm that to put together both the solid data and the 454 data actually improves very much the quality of the assembly. So I want to go now on another very related topic. So the message here is if we use this very powerful piece of information that is the mate pair, and if we use short reads, with the short reads, it's not so important the extension. It's important that you can anchor the two reads on something, on the contig that you have already produced. And what you use is essentially the information that allows you to distance properly all the contig. Another problem, instead, is to work out structural information, structural variation, sorry. So, structural variation are important both if you do resequencing. Resequencing means to find all the SNP of a certain individual, but also to find all the structural variation that we know are many structural variation that differentiate one genome from another of the same species. So, though the same approach, structural variation, is also relevant when you do de novo sequencing, because also with de novo sequencing, if you have a contig that is misassembled, then you can use the same program that you use for structural variation to see the misassembly. in a certain way, a misassembled contig is like if you have a structural variation inside your contig, and you can identify that exactly with the same program. This is a review that came out last year about comparing different methods for structural variation. We experimented some of them, but most of them are quite incomplete, and also they're very, very slow, and they have a very, very low sensitivity. So we try to dirty our hands also on this problem, and we try to develop a system to help. Just to remind you, what can we do with a mate pair signature? So if you have, in this case, an insertion, of course, the average length of the inserts that cover a certain position will be, if you have an insertion, it will result shorter because you expect something that is shorter. So the same thing if you have a deletion. And the nice thing also, if you have an inversion, you can detect it in a double way, because you can detect that at the end of the insertion the length is not right, but even more important, the direction of the reads is not right. So using the information that you get from the mate pair, you can identify these three situations that are essentially the base of structural variation. So what is the cribing approach? What we tried to do was to do a double approach. Since the sensitivity of all the available methods is very low, what we tried to do was to accept, to lower as much as possible the stringency. So that means that we have a better sensitivity, but we have to pay with a low specificity. So we have a lot of false positive if we lower the specificity. But to make things right, we added a second approach that other people don't do. So the first step is essentially a step like other program use. But then what we try to do is to go at the single base level and try to, if there is an insertion, for instance, we should find that some of the reads are split in that insertion and aligned partially on that position, and another piece of the read should align at the end of the insertion. So essentially what we did, we used the same program that we developed to align split read, to align, spliced read, to align transcripts on the genome. A lot of transcripts of the genome align on splicing side, and our PASS program is able to align them anyway and align a part of the reads on one side and another part at the other end of the intron. Well, we adapted this same protocol to and in practice, we consider an insertion as if it should be an intron, and very similarly, we can detect also the other kind of problems. So we have 4 indexes that we work out from the mate pair. So these 4 indexes then are plotted on a GBrowse, and you can see how they behave along the genome, and how they behave when you have an insertion or a deletion. To make easy the whole approach, we artificially inserted or deleted piece of the genome, and we can see how the program performs. So we have one index that is that consider mate pairs that are at the right distance, or not right distance, at an acceptable distance, and we want to see if the acceptable distance considered the single read is also confirmed by the all reads together. And sometimes if you have 100 reads, you have a significant indication that there is a small deletion or a small insertion. Instead, if you have a wrong distance and right orientation, this means that the mate pair are outside the possible range. So we know that there is definitely something wrong there, and probably there is a big insertion or a big deletion that make the mate pair completely out of range. Or another one is wrong orientation, and this is for inversion. And another possibility that we consider is that one mate pair aligns on one side, on one point, and the other mate pair doesn't align. And this is when you have an insertion of a new sequence that is not present. Of course, a deletion, one of the two ends doesn't go. The second step, as I said, is to go back to the putative region of misassembly and to try to align the sequence as if they were introns or... and this is the signal that you see. For instance, this is the physical coverage, so all the coverage, all the mate pair that cover that point. The yellow instead is the sequence coverage. But if you have an insertion, you can see very easily that here it goes down to 0, and therefore here there is something absolutely wrong. As I said, we keep a stringency very low, and we have false positive, though in cases like this it's quite clear anyway. But going back to the second stage, that is this at the end here, is very, you can see exactly the single base where the insertion was, because you can find some of the reads that are going across the whole thing. Short deletion is the same, so that was a long deletion. This is a short deletion where you apply a little bit of statistics, but the signal, this signal goes absolutely above the level, and again you can map it to the single base. Here is an insertion. Also with insertion you have similar signals, so you combine different signals to detect exactly what is going on, and this is inversion plus a small deletion, etc. The final message is that using this system we can identify very easily structural variation that are very difficult to find with a high fidelity, and we can go exactly to the single base, and so this is how our program performs. Of course, it depends very much on the coverage. If you have a 40x coverage, that now is quite acceptable, you have a very, very high rate of detection. Though this, now we have to redo this table because this was done with random insertion, deletion, and inversion, and some of them are very, very short. So when they are very short, you don't pick them up with this method. And so we'll have a new version of this table showing also the length of the structural variation. How many minutes do I have still?
5 minutes.
5 minutes. Let's try to make it in 2 minutes then, because we're all hungry. So let's go to the very final point. The very final point is, okay, very good. We have done all the production of data. We also managed to put all the data into a nice finished sequence, and now we want to make sense of the data. And make sense of the data is not very easy. And the typical problem, since I am in the session of human, right? And when you have a human genome and you have a genetic disease, well, you know that there is a genetic disease because there is a familiarity. Now, a possible approach, we tried it ourselves, is to resequence the whole genome and try to find what is going on. What happens is a nightmare, because what happens is that you find millions of alterations. It's very difficult to identify if the haplotypes, sometimes you don't know if two alleles are on the same strand or not. So there are a huge amount of problems. And a lot of mutation produce big problems, others produce little problems. So what do we do to answer this question? As now, my opinion is that you cannot click a button and say, okay, I can give you the answer. maybe in some very special cases you can, but in most cases you have to make guesses and try to get a help from the computer to find what is going wrong. So what do? We have produced a query platform where you can put, first of all the information is filtered by our programs. So each SNP, for instance, has a weight that depends on the effect that has on the protein, if it is a coding region, or if it is a UTR, or if it is a conserved region compared to other genomes and so on. So at the end of this story, you have a lot of weight that you can give to every single mutation. And then you can insert in your query system other things. For instance, if you're interested in a certain disease, you can put the gene ontology that is related to that disease. You can put, for instance, a genomic region, a certain chromosome, or even a more focused region. And all this information is put into the system. And most of the time, there is an answer that says, yes, I found something that satisfied all your questions. So what the computer returns is a ranked list of genes. So every gene has a point that depends on the mutation that has accumulated, on the gene ontology that you would like, on the chromosomal position, and so on. And at the end, you have this ranked list. But this takes only a few seconds. So if you don't like it and you want to change your hypothesis, you can change it. So with this interactive system, probably it will be easier to have an answer. To tell you the truth, we so far tried to resequence one genome. That was done more than 1 year ago with a coverage that wasn't too high, and we are still we haven't still found the final gene, but we have several hypotheses that we want to check, but it's certainly a long way to go. So the $1 million answer is still a very true thing. So this is my group, part of genomics and parts of bioinformatics, and thank you for your attention.
Thank you very much, Giorgio, for your enthusiastic presentation. Question? Please, Niels.
Thank you for this very interesting talk. You showed that next-gen sequencing maybe has replaced the CGH arrays for looking after structural variants. But does this apply to every genome, or do you need a very good reference sequence? Yeah.
Well, first of all, how can you do GCAs with an unknown genome? Or, I mean,
by next-gen sequencing, you can generate the
yes.
exome landscape of every genome.
That, this has more chance to apply for a non-well-characterized genome. The problem, if you want to do comparative genome hybridization, you need to compare at least two, and possibly at least one should be a bit more of a reference. Otherwise, you have to de novo sequencing with all the problems just to tell you something. With the tomato genome that we are finishing now to assemble, and this is an international team, it is now more than 1 year that we are struggling to put together the sequences, and that is with more than 20x coverage of 454 reads, 40x coverage of solid reads, and at least as many of Illumina reads. And even so, it's taking so long. So de novo sequencing, as I said, to do the novo assembly, at least you go to Peking, where they apparently can do it very easily, is still quite a problem. So I would say if you have a reference genome of your organism that you like, with a very reasonably low cost, you can certainly do a very good analysis of structural variation by mapping the mate pair that you produce on the reference, But I would say now I would recommend that at least one of the you have a reference, yes.